Long before the first Tyrannosaurus rex shook the ground, long before Triceratops grazed ancient plains, Earth belonged to creatures stranger and more terrifying than most people imagine. For nearly 300 million years, a span longer than the entire reign of the dinosaurs, the planet teemed with armored predators possessing jaws strong enough to crush bone, millipedes the size of cars, dragonflies with wingspans rivaling modern hawks, and saber-toothed killers that hunted with calculated ferocity. These weren't mythical beasts or fictional monsters. They were real animals that dominated Earth's oceans, lakes, swamps, and forests during the Paleozoic Era, from roughly 541 to 252 million years ago. The Cambrian seas witnessed the birth of complex predation. The Devonian oceans harbored armored fish larger than great white sharks. The Carboniferous forests sheltered arthropods of nightmarish proportions. And the Permian plains trembled under the footfalls of mammal-like predators with sabers longer than steak knives. This is their story, the untold chronicle of Earth's first super-predators. Witness 3 hours of deep-time terror, beauty, and resilience. Stay with us as we journey through the Paleozoic, the age that built the blueprint for all life on Earth. 635 million years ago, Earth emerged from one of the most extreme events in planetary history. The Marinoan glaciation had locked the world in ice for millions of years, a frozen nightmare that extended nearly to the equator. But as volcanic carbon dioxide accumulated in the atmosphere and greenhouse warming finally overpowered the albedo effect, the ice retreated. What followed was the Ediacaran period, a geological interval lasting until 539 million years ago, and with it came an astonishing development. For the first time in Earth's history, complex multicellular animals appeared. These were not hunters or prey in any sense we would recognize. They had no eyes, no mouths, no claws or teeth. Instead, they lived as passive absorbers and filter, feeders on a seafloor blanketed by bacterial carpets, an ecosystem so alien that scientists struggled for decades to determine whether some of these creatures were even animals at all. The Ediacaran oceans were unlike anything that exists today. Oxygen levels, though rising, remain dramatically lower than modern concentrations, perhaps reaching just 20 to 25% of present atmospheric levels by the early Ediacaran. The shallow seas may have held dissolved oxygen, but deeper waters remained largely anoxic, toxic with sulfur and iron compounds that would poison most life forms we know. Yet this was precisely the world in which Earth's first large, soft-bodied organisms took hold. Recent research suggests these early animals might have originated not in the sunlit shallows, as once assumed, but in the deep, cold, and crucially stable temperatures of the ocean slopes and basins. In these stenothermal environments, places where temperature fluctuated minimally, the synergistic stresses of low oxygen and thermal variation were reduced. Cold water is more viscous and makes oxygen harder to extract. But it also doesn't vary wildly, and for fragile, simple body plans operating near physiological limits, that stability may have been a refuge. The fossil record supports this hypothesis. The earliest Ediacaran animals appear around 571 million years ago in deep water facies, only moving into shelf environments roughly 555 million years ago. The event that seeded this diversification has been termed the Avalon Explosion, named after the Avalon Peninsula in Newfoundland, where some of the oldest and most exquisite fossils were discovered. Proposed in 2008 by Virginia Tech paleontologists, the Avalon Explosion predates the more famous Cambrian explosion by about 33 million years. It represents a rapid increase in metazoan biodiversity, including the first appearance of cyanidarians and bilaterians, fundamental branches on the animal tree of life. Unlike the Cambrian creatures that would follow, Avalon fauna were predominantly sessile, rooted to the seafloor like bizarre gardens. Many had frond-like or disc-shaped bodies, and they lived in communities that left stunning imprints in fine-grained sediments. The Avalon assemblage gives us our first glimpse of animals experimenting with size and structure, pushing the boundaries of what multicellular life could become. Among the most intriguing members of this ancient biota was Dickinsonia, an oval segmented organism that could grow up to 1.4 meters in length. For decades, scientists debated whether Dickinsonia was an animal, a giant protozoan, a fungus, or perhaps a lichen. The controversy stemmed from its utterly strange morphology: a flat, quilted body with repeated segments radiating from a central axis, no obvious head or appendages, and no discernible mouth or gut. In 2018, a breakthrough came when Russian doctoral student Ilya Bobrovsky carefully extracted organic material from 558 million-year-old Dickinsonia fossils preserved in the White Sea region of Russia. Analysis revealed that the creature contained 85 to 93% cholesterol, a lipid biomarker strongly associated with animal cells. The surrounding rock matrix, by contrast, was dominated by algal steroids. This chemical fingerprint provided the most compelling evidence yet that Dickinsonia was indeed an animal, likely related to modern placozoans and early metazoans. But how did it feed? Recent studies examining gut contents found that Dickinsonia specimens lacked traces of dietary molecules, suggesting they absorbed nutrients externally through their ventral surface, perhaps by grazing on microbial mats that covered the seafloor like living carpets. The Ediacaran seafloor was indeed dominated by extensive microbial mat communities, dense layers of cyanobacteria and other microorganisms that formed slimy, cohesive surfaces. These mats played a crucial role not only in the ecology of the time, but also in the preservation of Ediacaran fossils. Because the mats stabilized sediments and created sticky surfaces, they allowed the soft bodies of these creatures to leave detailed impressions when buried by volcanic ash or sand. In many cases, rapid entombment was followed by pyritization, or precipitation of silica, which replicated the organism's outlines in 3 dimensions. This mode of preservation, termed Ediacaran type, is extraordinarily rare in the fossil record and gives us an unusually clear window into these ancient communities. Without predators to tear bodies apart, or bioturbators to churn the sediment, both hallmarks of later Cambrian ecosystems, Ediacaran animals could rest undisturbed until geological processes immortalized them. Another iconic member of the Ediacaran world was Charnia, a frond-like organism anchored to the seafloor by a holdfast structure. Charnia belonged to the rangeomorphs, a group characterized by fractal, self-similar branching patterns. Standing upright in the water column, Charnia could reach heights of 60 centimeters or more, its feather-like branches extending outward to maximize surface area. Recent developmental studies using X-ray computed tomography have revealed that Charnia grew through successive branching, with new branches deriving from pre-existing ones in a hierarchical pattern. Remarkably, the organism had no central stalk. Instead, the entire frond was filled with interconnected branching units. This architecture is unlike anything alive today, leading researchers to classify Charnia as a stem eumetazoan, an early experiment in animal body plans that predates the divergence of cnidarians and bilaterians. The rangeomorphs thrived in deep water environments, often forming dense gardens, where individuals stood side by side in what may have been reproductive clusters. Perhaps the most convincing evidence for true bilateral animals in the Ediacaran comes from Kimberella, a slug-shaped organism found in rocks dating to 548 million years ago, near the White Sea. Kimberella measured between 3 and 150 millimeters in length and exhibited clear bilateral symmetry. Its fossils are frequently associated with clusters of paired scratch marks preserved in the sediment, traces interpreted as feeding marks created by a rasping organ similar to a molluscan radula. If this interpretation is correct, Kimberella represents not only one of the earliest bilaterians, but potentially an early mollusk or mollusk-like organism that actively grazed on microbial substrates. This would push back the divergence of major animal lineages, protostomes and deuterostomes, to well before the Cambrian boundary. Even paleontologists who dispute Kimberella's exact classification generally accept it as at least a bilaterian, making it a critical calibration point for molecular clock studies tracing animal evolution. The Ediacaran world, for all its strangeness, was remarkably peaceful compared to what would follow. There were no jaws, no claws, no shells built for defense. Predation, if it existed at all, was likely rare and primitive. But this tranquil age was not destined to last. As the Ediacaran drew to a close, diversity began to plummet. Paleontologists divide the Ediacaran biota into 3 successive assemblages based on their fossil occurrences. The Avalon assemblage, around 575 to 560 million years ago, the White Sea assemblage from 560 to 550 million years ago, and the Nama assemblage lasting from 550 to 539 million years ago. The White Sea assemblage was the most diverse, containing a rich variety of frondose rangemorphs, discoidal forms like Dickinsonia, and other enigmatic creatures. But when scientists examine the younger Nama assemblage, they find something alarming. Approximately 80% of the genera present in the White Sea strata are absent. This was no gradual fade. It was Earth's first known mass extinction. For decades, researchers debated what killed the Ediacaran animals. 2 major hypotheses emerged, termed the catastrophe model and the biotic replacement model. The catastrophe model proposed that environmental perturbations, perhaps shifts in ocean chemistry marked by negative carbon isotope excursions, triggered widespread die-offs similar to later Phanerozoic mass extinctions. The biotic replacement model, by contrast, suggested that the Ediacaran organisms were outcompeted and destroyed by newly evolving Cambrian-type animals that brought fundamentally new behaviors to the seafloor. These new creatures included mobile, bilaterians capable of burrowing into sediments, behavior known as bioturbation, which disrupted the microbial mats that Ediacaran organisms depended on and altered nutrient, cycling in ways that destabilized existing communities. Recent geochemical analyses have tipped the scales toward an environmental cause. In 2022, a team led by Scott Evans from Virginia Tech examined fossils from 4 sites spanning the terminal Ediacaran, including specimens from southern Namibia's Nama group. They documented dramatic shifts in body size, ecological gild composition, and morphological complexity across the extinction boundary. Crucially, the organisms that disappeared were disproportionately those with high oxygen requirements, large-bodied forms, and those with metabolisms suggesting active feeding or respiration. Geochemical proxies, including nitrogen isotopes and trace metal concentrations, indicated that ocean oxygenation fluctuated wildly during this interval, with sharp declines in dissolved oxygen coinciding with the extinction pulses. These findings suggest that the Ediacaran extinction, like all 5 major Phanerozoic mass extinctions, was environmentally driven. Yet the story is not one of simple replacement or catastrophe alone, but rather a complex interplay of both forces. As oxygen levels seesawed, new animals with novel capabilities, including the ability to burrow deeply into sediment, were radiating rapidly. The trace fossil record reveals that by the latest Ediacaran and earliest Cambrian, a diverse suite of burrowing behaviors had already appeared, including vertical shafts, horizontal tunnels, and complex 3-dimensional networks. These bioturbating animals fundamentally restructured benthic ecosystems, churning sediments, oxygenating deeper layers, and breaking up the cohesive microbial mats that had blanketed the seafloor for hundreds of millions of years. For sessile mat dwellers like Charnia, or passive absorbers like Dickinsonia, This new regime may have been as lethal as any chemical crisis. The microbial carpets that preserved their fossils so beautifully were themselves disappearing, and with them, the ecological niches these strange pioneers had occupied. By the dawn of the Cambrian, 539 million years ago, the Ediacaran biota had effectively vanished. A few stragglers persisted briefly into the earliest Cambrian, but they were ecological ghosts, the last remnants of a body-plan experiment that had run its course. What came next would be faster, fiercer, and unrecognizable to the soft gardens of the Ediacaran seas. 541 million years ago, Earth crossed an invisible threshold. The Cambrian period began not with a whisper, but with a biological detonation so rapid and so comprehensive that it remains one of the most profound mysteries in evolutionary science. Within a span of just 20 to 25 million years, a geological eye blink, nearly every major animal body plan known today appeared in the fossil record. Arthropods with jointed legs and compound eyes, mollusks with coiled shells, echinoderms with radial symmetry, and even the first chordates, our own distant ancestors, all emerged during this explosive interval. This was not gradual descent with modification. It was innovation on a scale never repeated. The event reshaped not only the oceans but the very rules of existence, introducing predation as a dominant ecological force and triggering an evolutionary arms race that would define all life to follow. The suddenness of the Cambrian explosion has puzzled scientists since Charles Darwin first acknowledged it as a potential challenge to his theory of evolution by natural selection. If animals evolved slowly over deep time through incremental changes, why does the fossil record show such an abrupt burst of complexity at the base of the Cambrian? Modern research suggests the answer lies not in a single cause, but in a cascade of interrelated factors, geological, geochemical, and biological, that converged at precisely the right moment to unlock an unprecedented wave of evolutionary experimentation. A 2013 study, led by Professor Paul Smith of Oxford University and Professor David Harper of Durham University, examined fossils from Sirius Passet in northern Greenland, just 500 miles from the North Pole, and concluded that the explosion resulted from multiple interacting processes rather than any standalone trigger. Their findings, published in the journal Science, described a sequence beginning with an early Cambrian sea level rise that dramatically increased the area of shallow continental shelf habitat available for colonization. This expansion in habitable space created ecological opportunities that had never existed before, providing niches for new body plans and lifestyles to flourish. But space alone was not sufficient. Geochemical shifts played an equally critical role. Atmospheric and oceanic oxygen levels, which had been rising fitfully since the Great Oxidation event more than 2 billion years earlier, reached a critical threshold by the early Cambrian. Estimates suggest that oxygen concentrations may have approached 15 to 20% of modern levels, still far below today's 21%, but high enough to support active mobile animals with higher metabolic demands. Oxygen is essential for energetically expensive processes, like building mineralized skeletons, powering muscle contractions, and supporting complex sensory organs. Without sufficient oxygen, large, active predators and heavily armored prey simply could not exist. The end of the last, Neoproterozoic glaciation, sometimes called the Gasquias glaciation around 580 million years ago, may have contributed to this oxygenation by releasing nutrients into warming oceans, fueling photosynthetic blooms that pumped oxygen into the water and atmosphere. Another key innovation was the widespread appearance of biomineralization, the ability to construct hard body parts from calcium carbonate, calcium phosphate or silica. Before the Cambrian, animals were almost exclusively soft-bodied, vulnerable, and limited in size. But as oceanic calcium concentrations increased, likely due to weathering of continental rocks and tectonic shifts, animals began precipitating minerals to form shells, plates, spines, and skeletal supports. These structures served multiple functions: protection from newly evolving predators, structural scaffolding to support larger body sizes, and attachment points for muscles that enabled more powerful and precise movements. The fossil record reflects this transformation vividly. The first small shelly fossils appear in earliest Cambrian rocks, quickly followed by fully developed exoskeletons and mineralized tests. This innovation was not confined to a single lineage. It evolved independently in multiple groups, from brachiopods to mollusks to arthropods, suggesting that the environmental conditions strongly favored hard parts across the board. Perhaps the most transformative development was the evolution of vision. Eyes, specifically compound eyes capable of forming images, gave animals the ability to detect predators, locate prey, and navigate complex environments with unprecedented precision. The emergence of predation fundamentally restructured ecological relationships, shifting life from passive filter feeding and grazing toward active hunting and evasion. This ignited a co-evolutionary arms race. Predators evolved sharper claws, stronger jaws, and faster locomotion, while prey responded with defensive armor, camouflage, and burrowing behaviors. The fossil record captures this escalation beautifully. The diversity of trace fossils, which record animal activity rather than bodies, explodes at the Cambrian boundary, showing an array of burrowing depths, tunnel architectures, and locomotory patterns far exceeding anything from the Ediacaran. The window into this ancient explosion comes from a handful of extraordinary fossil deposits where soft tissues were preserved in exquisite detail, something that almost never happens in the geological record. The most famous of these is the Burgess Shale, located high in the Canadian Rockies of British Columbia. On August 30th, 1909, paleontologist Charles Doolittle Walcott of the Smithsonian Institution made a discovery that would revolutionize our understanding of early animal life. According to legend, though likely apocryphal, Walcott's horse stumbled on a loose rock, and when he examined it, he found the imprint of a soft-bodied creature unlike anything he had ever seen. Whether or not the story is true, Walcott recognized the significance immediately. He established a quarry on the flanks of Fossil Ridge and returned nearly every summer until 1924, amassing over 65,000 specimens by the time he was 74 years old. He spent the rest of his life describing these fossils, but died in 1927 before completing the work. Walcott's interpretations, however, were constrained by the scientific paradigms of his era. He attempted to fit every Burgess Shale organism into existing taxonomic groups, classifying them as primitive versions of modern animals. As a result, the true significance of the fossils remained hidden for decades, and they were largely dismissed as curiosities. It was not until 1962, when Italian paleontologist Alberto Simonetta conducted a first-hand reinvestigation, that scientists began to appreciate what they actually had. In the 1970s and 1980s, a team led by Harry Whittington at Cambridge University, along with his graduate students, Derek Briggs and Simon Conway Morris, undertook a comprehensive reanalysis of the Burgess Shale fauna. What they found was astonishing. Many of the organisms could not be shoehorned into modern phyla at all. Some possessed body plans so strange, so unlike anything alive today, that they seemed to represent entirely extinct experiments in animal architecture. When Whittington first presented his reconstruction of Opabinia, a creature with 5 eyes on stalks and a frontal grasping appendage like an elephant's trunk, the audience reportedly laughed, assuming he had made a mistake. The Burgess Shale fauna revealed just how wildly diverse Cambrian animals were. Wiwaxia, for instance, was a slug-like creature covered in rows of protective carbonaceous scales and spines, arranged in a bilaterally symmetrical pattern across its oval body. Measuring just a few centimeters long, Wiwaxia crept slowly across the seafloor, using a toothed feeding apparatus on its underside to scrape bacteria and microorganisms from microbial mats. Its sclerites, hardened scales, were individually added and shed throughout growth. maintaining body symmetry in a manner that suggested a possible molluscan affinity, specifically resembling the eightfold segmentation seen in modern chitons. Remarkably, Wiwaxia fossils have been found not only in the Burgess Shale, but also in deposits from China, Australia and the Czech Republic, indicating that this body plan was widespread and successful across Cambrian seas. Yet, despite its abundance, Wiwaxia disappeared entirely, leaving no direct descendants. Even stranger was Hallucigenia, whose name aptly captures the bewilderment it induced in researchers. This worm-like animal had a long tubular body bearing 2 rows of tall dorsal spines along its back and pairs of clawed legs beneath. When first reconstructed, scientists placed it upside down and backward, a testament to how alien its anatomy was. Only after re-examination of additional specimens did the true orientation become clear. The spines were defensive structures, and the legs, tipped with claws, allowed it to walk along the seafloor. Hallucigenia belonged to a group called Lobopodians, which are considered relatives or precursors of modern velvet worms and arthropods. Its discovery underscored a key lesson of the Burgess Shale. Cambrian animals were not simply primitive versions of modern forms, but rather participants in a vast evolutionary experiment, many branches of which led nowhere. The diversity of body plans, seen in the Burgess Shale and equivalent deposits like China's Chengjiang fauna, reflects the unparalleled morphological freedom of the early Cambrian. Nearly all major animal phyla, Arthropoda, Mollusca, Echinodermata, Chordata, Brachiopoda, Cnidaria, Porifera, and Priapulida, made their first appearance during this narrow window. The Cambrian explosion established the basic blueprints for animal life, blueprints that have remained largely unchanged for over 500 million years. Later radiations, such as the diversification of fish in the Silurian and Devonian, or the recovery after the Permian-Triassic extinction, involved far fewer fundamentally new body plans. The ecological niches were already occupied, and competition was fierce. The Cambrian was unique because it was a time of empty space, open water columns, undefended seafloors, and no established predators. Life rushed in to fill every available niche, and in doing so, set the template for all animal ecosystems to come. Among the most remarkable ecological innovations of the Cambrian was the emergence of the first metazoan reef ecosystems, built not by corals, which would not evolve for another 100 million years, but by bizarre cup-shaped organisms called archaeocyaths. These sponge-grade animals first appeared around 530 million years ago in what is now Siberia during the Timoteian Age, and rapidly spread across warm, shallow tropical seas worldwide. Archaeocyaths had calcified skeletons with double walls perforated by pores, and they anchored themselves to hard substrates, growing upward into structures that provided the framework for complex reef communities. Their ecological role was transformative. By creating 3-dimensional habitats with cavities, overhangs, and hard surfaces, archaeocyath reefs offered shelter and attachment sites for a diverse array of other organisms, including trilobites, brachiopods, echinoderms, and early mollusks. Recent computational fluid dynamics simulations have demonstrated that archaeocyaths were active suspension feeders, pumping water through their central cavities to capture organic particles, rather than relying passively on ambient currents. This active feeding strategy, combined with their ability to biomineralize, allowed them to thrive in environments ranging from high-energy wave-swept crests to deeper, quieter reef mounds. The ability to construct massive calcified skeletons depended critically on ocean chemistry, which was undergoing profound changes during the Cambrian. Increased weathering of exposed continental rocks delivered unprecedented quantities of calcium and other ions into the oceans. Evidence for this comes from carbonate rocks laid down during the Cambrian, which contain roughly 3 times as much calcium carbonate as typical marine sediments from other periods. Magnesium to calcium ratios in seawater fluctuated through the Cambrian, producing a complex succession of aragonite seas, mixed aragonite calcite seas, and eventually calcite seas. During Cambrian Stage 2, Aragonite and high magnesium calcite were the dominant precipitates. But by the Middle Cambrian, conditions had shifted toward more calcite-rich mineralization. These chemical shifts influenced which organisms could successfully biomineralize and what kind of skeletons they produced. Importantly, rising oxygen levels reduced ocean acidity, increasing carbonate saturation, and making it energetically easier for organisms to precipitate calcium carbonate shells and skeletons. This combination of abundant dissolved calcium, favorable pH conditions, and sufficient oxygen created a perfect storm for the evolution of hard parts across multiple lineages simultaneously. Trilobites became perhaps the most successful and diverse group of Cambrian arthropods, rapidly radiating into hundreds of genera adapted to nearly every marine environment. These segmented creatures had compound eyes, jointed legs, and mineralized exoskeletons made of calcite that could be molted as they grew. The earliest trilobites appeared in the Lower Cambrian and quickly diversified into major orders including Redlichida, Tychoparida, Agnostida, and Coronexochida. A significant crisis struck trilobite populations during the Middle Cambrian, and surviving lineages responded by evolving thicker, more heavily armored cuticles and larger tail shields, adaptations likely driven by increasing predation pressure. The Late Cambrian witnessed the apex of trilobite diversity, with species occupying reef environments, open shelves, and deeper basins. But this golden age was short-lived. The End Cambrian mass extinction event, around 488 million years ago, devastated trilobite faunas. Almost all members of the Redlichida, including the once dominant Olenelloidea, were wiped out, and most late Cambrian stocks disappeared. This extinction coincided with major environmental upheaval, including a contraction of shallow continental shelf areas, fluctuations in sea level, and disruptions to ocean chemistry and circulation patterns. The window into this ancient explosion comes from a handful of extraordinary fossil deposits where soft tissues were preserved in exquisite detail, something that almost never happens in the geological record. The most famous of these is the Burgess Shale, located high in the Canadian Rockies of British Columbia. On August 30th, 1909, paleontologist Charles Doolittle Walcott of the Smithsonian Institution made a discovery that would revolutionize our understanding of early animal life. According to legend, though likely apocryphal, Walcott's horse stumbled on a loose rock, and when he examined it, he found the imprint of a soft-bodied creature unlike anything he had ever seen. Whether or not the story is true, Walcott recognized the significance immediately. He established a quarry on the flanks of Fossil Ridge and returned nearly every summer until 1924, amassing over 65,000 specimens by the time he was 74 years old. He spent the rest of his life describing these fossils, but died in 1927 before completing the work. Walcott's interpretations, however, were constrained by the scientific paradigms of his era. He attempted to fit every Burgess Shale organism into existing taxonomic groups, classifying them as primitive versions of modern animals. As a result, the true significance of the fossils remained hidden for decades, and they were largely dismissed as curiosities. It was not until 1962, when Italian paleontologist Alberto Simonetta conducted a first-hand reinvestigation, that scientists began to appreciate what they actually had. In the 1970s and 1980s, a team led by Harry Whittington at Cambridge University, along with his graduate students Derek Briggs and Simon Conway Morris, undertook a comprehensive reanalysis of the Burgess Shale fauna. What they found was astonishing. Many of the organisms could not be shoehorned into modern phyla at all. Some possessed body plans so strange, so unlike anything alive today, that they seem to represent entirely extinct experiments in animal architecture. When Whittington first presented his reconstruction of Opabinia, a creature with 5 eyes on stalks and a frontal grasping appendage like an elephant's trunk, the audience reportedly laughed, assuming he had made a mistake. The Burgess Shale fauna revealed just how wildly diverse Cambrian animals were. Waiwaxia, for instance, was a slug-like creature covered in rows of protective carbonaceous scales and spines arranged in a bilaterally symmetrical pattern across its oval body. Measuring just a few centimeters long, Waiwaxia crept slowly across the seafloor, using a toothed feeding apparatus on its underside to scrape bacteria and microorganisms from microbial mats. Its sclerites, hardened scales, were individually added and shed throughout growth, maintaining body symmetry in a manner that suggested a possible molluscan affinity, specifically resembling the eightfold segmentation seen in modern chitons. Remarkably, Waiwaxia fossils have been found not only in the Burgess Shale, but also in deposits from China, Australia, and the Czech Republic, indicating that this body plan was widespread and successful across Cambrian seas. Yet despite its abundance, Waiwaxia disappeared entirely, leaving no direct descendants. Even stranger was Hallucigenia, whose name aptly captures the bewilderment it induced in researchers. This worm-like animal had a long tubular body, bearing 2 rows of tall dorsal spines along its back and pairs of clawed legs beneath. When first reconstructed, scientists placed it upside down and backward, a testament to how alien its anatomy was. Only after re-examination of additional specimens did the true orientation become clear. The spines were defensive structures, and the legs, tipped with claws, allowed it to walk along the seafloor. Hallucigenia belonged to a group called Lobopodians, which are considered relatives or precursors of modern velvet worms and arthropods. Its discovery underscored a key lesson of the Burgess Shale. Cambrian animals were not simply primitive versions of modern forms, but rather participants in a vast evolutionary experiment, many branches of which led nowhere. The diversity of body plans, seen in the Burgess Shale and equivalent deposits like China's Chengjiang fauna, reflects the unparalleled morphological freedom of the early Cambrian. Nearly all major animal phyla, Arthropoda, Mollusca, Echinodermata, Chordata, Brachiopoda, Cnidaria, Porifera, and Priapulida made their first appearance during this narrow window. The Cambrian explosion established the basic blueprints for animal life, blueprints that have remained largely unchanged for over 500 million years. Later radiations, such as the diversification of fish in the Silurian and Devonian, or the recovery after the Permian-Triassic extinction, involved far fewer fundamentally new body plans. The ecological niches were already occupied, and competition was fierce. The Cambrian was unique because it was a time of empty space, open water columns, undefended seafloors, and no established predators. Life rushed in to fill every available niche, and in doing so, set the template for all animal ecosystems to come. Among the most remarkable ecological innovations of the Cambrian was the emergence of the first metazoan reef ecosystems, built not by corals, which would not evolve for another 100 million years, but by bizarre cup-shaped organisms called archaeocyaths. These sponge-grade animals first appeared around 530 million years ago in what is now Siberia during the Timoteian Age, and rapidly spread across warm, shallow tropical seas worldwide. Archaeocyaths had calcified skeletons with double walls perforated by pores, and they anchored themselves to hard substrates, growing upward into structures that provided the framework for complex reef communities. Their ecological role was transformative, by creating 3-dimensional habitats with cavities, overhangs, and hard surfaces. Archaeocyath reefs offered shelter and attachment sites for a diverse array of other organisms, including trilobites, brachiopods, echinoderms, and early mollusks. Recent computational fluid dynamics simulations have demonstrated that archaeocyaths were active suspension feeders, pumping water through their central cavities to capture organic particles, rather than relying passively on ambient currents. This active feeding strategy, combined with their ability to biomineralize, allowed them to thrive in environments ranging from high-energy wave-swept crests to deeper, quieter reef mounds. The ability to construct massive calcified skeletons depended critically on ocean chemistry, which was undergoing profound changes during the Cambrian. Increased weathering of exposed continental rocks delivered unprecedented quantities of calcium and other ions into the oceans. Evidence for this comes from carbonate rocks laid down during the Cambrian, which contain roughly 3 times as much calcium carbonate as typical marine sediments from other periods. Magnesium to calcium ratios in seawater fluctuated through the Cambrian, producing a complex succession of aragonite seas, mixed aragonite calcite seas, and eventually calcite seas. During Cambrian Stage 2, aragonite and high magnesium calcite were the dominant precipitates, but by the Middle Cambrian, conditions had shifted toward more calcite-rich mineralization. These chemical shifts influenced which organisms could successfully biomineralize and what kind of skeletons they produced. Importantly, rising oxygen levels reduced ocean acidity, increasing carbonate saturation, and making it energetically easier for organisms to precipitate calcium carbonate shells and skeletons. This combination of abundant dissolved calcium, favorable pH conditions, and sufficient oxygen created a perfect storm for the evolution of hard parts across multiple lineages simultaneously. Trilobites became perhaps the most successful and diverse group of Cambrian arthropods, rapidly radiating into hundreds of genera adapted to nearly every marine environment. These segmented creatures had compound eyes, jointed legs, and mineralized exoskeletons made of calcite that could be molted as they grew. The earliest trilobites appeared in the Lower Cambrian and quickly diversified into major orders including Redlichida, Tychoparida, Agnostida, and Coronexocida. A significant crisis struck trilobite populations during the Middle Cambrian, and surviving lineages responded by evolving thicker, more heavily armored cuticles and larger tail shields, adaptations likely driven by increasing predation pressure. The Late Cambrian witnessed the apex of trilobite diversity, with species occupying reef environments, open shelves, and deeper basins. But this golden age was short-lived. The End Cambrian mass extinction event, around 488 million years ago, devastated trilobite faunas. Almost all members of the Redlichida, including the once dominant Olaneloidea, were wiped out, and most late Cambrian stocks disappeared. This extinction coincided with major environmental upheaval, including a contraction of shallow continental shelf areas, fluctuations in sea level, and disruptions to ocean chemistry and circulation patterns. Anomalocaris was not alone in the Cambrian seas. It belonged to an entire order of stem group arthropods called Radiodonta, literally radial teeth, a reference to their distinctive circular mouth structures. At least 37 species of radiodonts have been identified from deposits spanning Cambrian Stage 3 through the early Ordovician, and possibly extending into the Lower Devonian with the enigmatic genus Schinderhannes, though this attribution remains controversial. These animals exhibited a remarkable range of body sizes, feeding strategies, and ecological niches, demonstrating that early arthropod predators were far more diverse and adaptable than once imagined. Herdia victoria, for instance, was a close relative of Anomalocaris, but possessed a massive head shield that extended backward over its body like a helmet, possibly serving as protection or hydrodynamic streamlining. Unlike the slender appendages of Anomalocaris, Herdia's frontal limbs bore thick, widely spaced auxiliary spines that were relatively short and robust. These anatomical differences suggest Herdia was a sediment sifter, using its appendages to rake through seafloor mud and extract buried prey such as worms and small arthropods. Even more specialized was Peitoia nathorsti, the very organism whose mouth structure had originally been misidentified as a jellyfish. Peitoia belonged to the Herdiid family and possessed short-stalked eyes positioned behind its large head, along with frontal appendages bearing long bristle-like spines rather than the robust grasping structures seen in Anomalocaris. It lacked a fan-shaped tail, suggesting a different mode of swimming or perhaps a more benthic lifestyle. Peitoia infracambriensis, from the Zawishan Formation of Poland, is now recognized as the geologically oldest known radiodont species, pushing the origin of this group back to Cambrian Stage 3. The discovery of multiple radiodont genera in single fossil assemblages such as the Margam Formation of Utah, which has yielded Peytoia, Caryocyntrips pavantia, and the newly described Bucaspinea cooperi, indicates that these predators coexisted by partitioning resources and occupying distinct ecological roles. Bucaspinea cooperi, described in 2021, possessed a large oral cone with robust hooked teeth bearing 1 to 3 cusps, and its frontal appendages had at least 12 segments bearing 6 recurved plate-like endites proximally, and up to 4 spiniform distal endites. This unique morphology suggests yet another feeding strategy, possibly targeting different prey types or sizes than its contemporaries. The global distribution of radiodonts underscores their ecological success during the Cambrian radiation. Anomalocaris canadensis fossils have been recovered not only from the Burgess Shale of British Columbia, but also from the Chengjiang biota of Yunnan province in China, the Emu Bay Shale of South Australia, the Kylie Formation of southern China, and the Wheeler and Weeks formations of Utah. The Chengjiang biota, dated to approximately 518 million years ago, roughly 10 million years older than the Burgess Shale, contains some of the most exquisitely preserved radiodont specimens ever discovered, including complete body fossils with soft tissues, internal structures, and even gut contents visible. These deposits represent diverse depositional environments, ranging from shallow tropical deltas to deeper offshore basins, demonstrating that radiodonts were not restricted to a single habitat type, but instead colonized a wide range of marine settings. Interestingly, herdide radiodonts like Herdia and Peytoia appear to have been rarer in the Chengjiang biota than in North American deposits, possibly due to differences in water depth, sedimentation rates, or ecological conditions that favored different radiodont clades in different regions. But despite their dominance throughout the Cambrian, radiodonts did not survive indefinitely. The group suffered significant losses during the Cambrian Ordovician extinction event approximately 485 million years ago, which eliminated many brachiopods, conodonts, and trilobite species. The extinction was likely driven by a combination of factors including volcanic activity, such as the emplacement of large igneous provinces in South Africa, persistent anoxic conditions in deeper waters, fluctuating sea levels, and possibly increased hydrogen sulfide concentrations in the oceans. Radiodonts, as large active predators with high metabolic demands, would have been particularly vulnerable to disruptions in oxygen availability and changes in prey abundance. A few radiodont lineages persisted into the Ordovician, including some miniaturized forms discovered in whales that lived in sponge-dominated habitats, far removed from the open water niches their Cambrian ancestors had occupied. These Ordovician survivors represent a final diminished chapter in radiodont history, and by the Silurian, the order had vanished entirely, replaced by new predatory arthropods including the fearsome Eurypterids, the sea scorpions, that would dominate the next phase of marine predation. The Burgess Shale and its equivalents across the globe preserve a menagerie of creatures so bizarre that when they were first reconstructed, scientists questioned whether they could have functioned at all. These were not merely primitive versions of familiar animals, but entirely novel experiments in animal architecture. Body plans that pushed the boundaries of what multicellular life could achieve. Among the strangest was Opabinia regalis, a creature that looked as though it had been assembled from spare parts belonging to different animals. Measuring about 3 inches long, Opabinia possessed 5 eyes arranged across the top of its head, like the lenses of a compound microscope. 4 of these eyes were positioned in a rectangular array, with a 5th smaller eye mounted centrally between them, giving the animal nearly panoramic vision and exceptional depth perception. But the most extraordinary feature was its feeding apparatus. A long flexible proboscis extending from the front of the head, tipped with a pair of grasping spines that could bend backward to deliver food to a mouth positioned on the underside of the head behind the proboscis. Base. When paleontologist Harry Whittington first presented his reconstruction of Opabinia at a 1972 scientific conference, the audience reportedly burst into laughter, assuming he had made an error. The animals seemed too improbable, too awkward to have actually existed. Yet subsequent discoveries confirmed every detail. Opabinia's body consisted of 15 segments, each bearing lateral lobes that overlapped like roof tiles and undulated to propel the animal through water. The lobes on the first body segment were enlarged and oriented differently from the rest, possibly functioning as steering structures. The tail terminated in 3 pairs of fan-shaped fins, arranged in a distinctive pattern that has no modern equivalent. Recent phylogenetic analyses place Opabinia as a stem group arthropod, related to but distinct from radiodonts, like Anomalocaris. Its unique anatomy suggests it occupied a specialized ecological niche, likely using its flexible proboscis to probe sediments and extract small invertebrates, worms, and organic debris from burrows and crevices that other predators could not access. Opabinia was far from the only oddity in Cambrian seas. The ocean floors teemed with creatures whose body plans defy easy classification even today. Marella splendens, nicknamed the lace crab, was the most abundant arthropod in the Burgess Shale, represented by over 25,000 specimens. This delicate animal possessed 2 pairs of long, curved spines projecting backward from its head shield, and dozens of feathery legs that it used for swimming and filter feeding. Despite its abundance and excellent preservation, Marella's exact evolutionary position remains uncertain. It shares features with both crustaceans and chelicerates, but belongs definitively to neither group. Nectocaris pteryx looked superficially like a squid with a streamlined body, lateral fins, long tentacles, and large eyes. Yet it lived more than 300 million years before the first true cephalopods evolved. For decades, scientists debated whether Nectocaris was a bizarre arthropod, an early mollusk, or something else entirely. Current evidence suggests it may represent a stem group cephalopod or a very early member of the mollusk lineage, demonstrating that the origins of major animal groups were far messier and more complicated than neat evolutionary trees suggest. Yet among all the Cambrian oddities, trilobites stand as the most successful and enduring. These arthropods first appeared around 521 million years ago at the beginning of Cambrian Stage 3 and quickly radiated into hundreds of genera that colonized every marine habitat from tropical reefs to deep ocean basins. The earliest trilobites were already anatomically complex, possessing fully segmented bodies divided into 3 longitudinal lobes: a cephalon or head shield, a thorax of articulated segments, and a pygidium or tail shield. But their most remarkable feature was their eyes. Even the oldest trilobites possessed sophisticated compound eyes constructed from calcite crystals, the only animals in Earth's history to evolve mineralized lenses. Each lens was an elongated prism of crystallographically oriented calcite, arranged hexagonally in arrays that could contain anywhere from a single lens to more than 15,000 individual units in a single eye. The optical sophistication of trilobite eyes is astonishing. Pure calcite is transparent, and trilobites exploited this property to create lenses with extraordinary clarity. In some lineages, the calcite formed an internal doublet structure, 2 layers with different refractive indices that corrected for spherical aberration and provided exceptional depth of field. This design follows optical principles that would not be formally described by humans until René Descartes and Christian Huygens worked them out in the 17th century. Fossils from the early Cambrian Emu Bay Shale of South Australia, dated to approximately 515 million years ago, preserve trilobite eyes in such exquisite detail that individual lenses can be examined under microscopy. These eyes contained over 3,000 large ommatidial lenses and featured a specialized bright zone, a region with larger, more densely packed lenses that enhanced sensitivity in specific directions. The arrangement and size of the lenses indicate these trilobites were active predators or scavengers capable of seeing in low light conditions. Remarkably, non-mineralized compound eyes of similar complexity do not appear again in the fossil record until about 85 million years later, making these the oldest and most advanced early eyes known from any animal. Some trilobites even possessed median eyes, simple photoreceptors positioned on the central axis of the head between the compound eyes. These median eyes, documented in multiple trilobite genera from Cambrian deposits, functioned as light detection organs that helped regulate circadian rhythms, detect shadows of approaching predators, or orient relative to sunlight for navigation. The presence of both compound and median eyes in the earliest trilobites confirms that sophisticated vision had evolved before the beginning of the Cambrian explosion, not during it, pushing the origins of animal eyes even deeper into the Ediacaran or earliest Cambrian. The rapid refinement and diversification of visual systems during the Cambrian has been proposed as one of the key evolutionary pressures driving the explosion itself. As predators evolved better eyes, prey evolved defensive behaviors, camouflage, and armor, igniting an escalating coevolutionary arms race that accelerated speciation across the board. By the middle to late Cambrian, around 500 million years ago, trilobites had reached their peak diversity, with thousands of species occupying ecological roles, ranging from burrowing deposit feeders to active swimmers to reef-dwelling omnivores. Their fossil record is extraordinarily rich, with over 22,000 described species spanning the entire Paleozoic era, making them one of the most diverse groups of extinct animals ever documented. This abundance and morphological variation have made trilobites invaluable biostratigraphic markers, allowing geologists to date and correlate rock layers across continents with precision. Their easily fossilized calcite exoskeletons, which they shed and regrew repeatedly as they grew, ensured that trilobite remains are common in marine sediments worldwide. But the Cambrian seas held one more surprise, perhaps the most significant for human history: the first chordates, animals possessing a notochord, dorsal nerve cord, and segmented muscles, the fundamental body plan that would eventually give rise to all vertebrates, including ourselves. Picaia gracilans, discovered by Charles Doolittle Walcott in the Burgess Shale and described in 1911, measured about 2 inches long and resembled a flattened eel. Walcott initially classified it as an annelid worm based on its transverse body segments. But in 1979, Harry Whittington and Simon Conway Morris reanalyzed the fossils and conclusively identified it as a chordate. The key evidence was a stiff rod running the length of the body, a notochord, and V-shaped muscle blocks called myomeres that allowed undulating, eel-like swimming. Picaia lacked a head, eyes, or jaws, and its body was covered by a cuticle similar to that of invertebrates, marking it as a very primitive member of the chordate lineage. Recent reinterpretations in 2024 found evidence of a gut canal and dorsal nerve cord and revealed that the animal had been reconstructed upside down for decades. Even more derived were Hycoichthys and Milocunmingia, tiny fish-like creatures from the Chengjiang biota of China, dated to approximately 518 million years ago. These animals, measuring just an inch or two in length, possessed all the hallmarks of stem vertebrates: a notochord, segmented muscles, gill slits, a distinct head with sensory organs, and possibly even rudimentary cartilaginous structures supporting the gills. Hycoichthys represents one of the earliest known vertebrates, animals with a backbone-like support system, bridging the gap between simple chordates like Picaia and true-jawed fish that would dominate later periods. Their discovery demonstrates that vertebrate origins trace directly back to the Cambrian explosion, meaning that our own evolutionary lineage was already present among the bizarre menagerie of Burgess Shale creatures over 500 million years ago. The extraordinary diversity of the Cambrian world did not last. Around 515 million years ago, midway through the period, something went drastically wrong. The number of species going extinct suddenly exceeded the number of new species appearing, and biodiversity plummeted. Within 5 million years, the number of genera had crashed from a peak of roughly 600 to just 450, a loss of a quarter of all diversity. The speciation rate in many groups fell to between 1/5 and 1/3 of previous levels. Then, around 500 million years ago, oceanic oxygen levels dropped precipitously, creating widespread hypoxic conditions in shallow marine environments. Simultaneously, concentrations of hydrogen sulfide, a deadly toxin to most aerobic life, spiked upward, triggering another round of extinctions. The late Cambrian was, contrary to popular imagination, a time of crisis and decline, marked by multiple rapid extinction pulses that winnowed the fantastic diversity of the earlier period, down to a fraction of its former richness, the Archaeocath reefs that had flourished. Across tropical shelves vanished entirely by the Middle Cambrian, replaced once again by microbial stromatolites, the ancient bacterial structures that had dominated before complex animals evolved. The return of stromatolites signaled a regression, an ecological simplification, brought on by the collapse of reef-building metazoans. Many of the bizarre body plans documented in the Burgess Shale, Opabinia, Wywaxia, Hallucigenia, and countless others, disappeared without leaving descendants. These were evolutionary dead ends, experiments that worked brilliantly for millions of years, but ultimately could not compete with more derived arthropods, mollusks, and other groups that refined their anatomies through successive generations. Priapulid worms, which were abundant and diverse in the Cambrian, suffered catastrophic losses and survive today only as a minor phylum restricted to cold water mud habitats, relics of a once dominant group. The final blow came at the Cambrian-Ordovician boundary, approximately 485 million years ago. This extinction event eliminated many brachiopod and conodont lineages and severely reduced trilobite diversity. Volcanic activity, particularly the emplacement of massive igneous provinces such as the Namaquel and Gary's dikes in South Africa, has been implicated as a potential cause. The eruptions would have released vast quantities of carbon dioxide and sulfur compounds, acidifying the oceans and disrupting marine ecosystems already stressed by anoxia and fluctuating sea levels. Recent geological studies of sites in Australia and Antarctica have revealed tectonic shifts during this interval that altered ocean circulation patterns and reduced the area of shallow continental shelf habitat, compressing species ranges and increasing extinction risk. The persistent anoxic conditions that characterized much of the Middle to Late Cambrian may have maintained chronically elevated extinction rates, preventing full recovery between crises. Trilobites, despite their sophistication and diversity, were among the hardest hit. The dominant families of the Late Cambrian, particularly the Redlichida and Olinelloidea, were almost entirely wiped out. Survivors faced a transformed world in the early Ordovician, one where competition and predation pressures were intensifying. Origination and extinction rates, which had fluctuated wildly throughout the Cambrian, dropped sharply after the boundary and stabilized at much lower levels through the Ordovician. This shift marked a fundamental change in evolutionary dynamics. The chaotic, high-turnover world of the Cambrian gave way to a more structured, competitive regime where ecological specialization became paramount. Trilobites that survived into the Ordovician developed thicker exoskeletons, defensive spines, and the ability to enroll, rolling into a ball like modern pillbugs, to protect themselves from predators. But they would never again achieve the morphological freedom or unconstrained diversity of their Cambrian heyday. The age of strange survivors had ended, and a new chapter was beginning, one dominated by predators far larger and more terrifying than anything Anomalocaris could have imagined. The Ordovician period, stretching from 485 to 443 million years ago, witnessed one of the most dramatic evolutionary radiations in Earth's history. Known as the Great Ordovician Biodiversification Event, this explosion of marine life saw the number of genera triple and the complexity of ecosystems increase beyond anything the Cambrian had achieved. Brachiopods diversified into hundreds of forms. Bryozoans constructed delicate branching colonies, and the first true corals began building reefs that would rival the lost Archaeocyath structures. Trilobites, survivors of the Cambrian crisis, evolved bizarre defensive adaptations. Some developed long eye stalks to watch for predators from a distance. Others grew elaborate spines and nodules as armor. and still others adopted a swimming lifestyle to escape benthic hunters. The arms race that began in the Cambrian was accelerating, and the oceans were filling with increasingly sophisticated predators. Among the most formidable were the eurypterids, arthropods that would come to be known as sea scorpions, though they were neither true scorpions nor confined exclusively to the sea. Eurypterids first appeared in the Middle Ordovician, around 467 million years ago, emerging from the same arthropod lineage that would eventually give rise to horseshoe crabs, spiders, and true scorpions. These early eurypterids were relatively small and unremarkable, measuring just a few inches in length and likely occupying minor predatory or scavenging roles in shallow coastal environments. But they possessed a body plan with extraordinary potential, a segmented exoskeleton that could be modified for speed and strength, paddle-like appendages capable of powerful swimming strokes, compound eyes mounted on a broad head shield for wide-angle vision, and most distinctively, a pair of specialized frontal appendages that could be adapted for grasping, crushing, or slashing prey. Unlike their distant relatives, the trilobites, which were constrained by their inflexible body architecture, eurypterids could evolve rapidly to exploit new niches. By the late Ordovician, just a few million years after their origin, eurypterids had already begun to diversify, and some lineages were experimenting with larger body sizes. The transition from the Ordovician to the Silurian period, beginning 443 million years ago, was marked by one of the most severe mass extinctions in Earth's history. The late Ordovician glaciation, triggered when the supercontinent Gondwana drifted over the South Pole, locked vast quantities of water in continental ice sheets and caused sea levels to plummet. Shallow marine habitats, where most life was concentrated, dried up or became inhospitable as temperatures dropped and ocean chemistry shifted. An estimated 85% of marine species disappeared in two pulses separated by just a few 100,000 years. Trilobites lost half their families, brachiopods suffered devastating losses, and the dominant graptolite groups that had filled Ordovician seas were nearly wiped out. But eurypterids, perhaps because of their tolerance for variable salinity and their ability to inhabit estuaries and brackish lagoons, survived the crisis with minimal losses. As global temperatures rebounded in the early Silurian and seas reflooded the continents, eurypterids underwent an explosive adaptive radiation that would make them the apex predators of the Silurian world. By the Silurian and early Devonian periods, eurypterids had evolved into apex predators of astonishing size. Pterygotus, a genus that would spread across 4 continents with a nearly cosmopolitan distribution, reached lengths of 1.6 to 1.75 meters depending on the species. The largest species, Pterygotus grandidentatus, stretched 5.7 feet from head to tail, with massive chelicerae, frontal claws, capable of puncturing and grasping prey with crushing force. These were not the delicate, grasping appendages of Anomalocaris. They were robust, heavily toothed pincers, more similar to the claws of modern lobsters and crabs, designed explicitly for active predation. The chelicerae were so large that a single claw could measure a foot in length, with teeth readily discernible even when partially embedded in rock matrix. Pterygotus may have weighed around 30 kilograms, a staggering mass for an arthropod, equivalent to a medium-sized dog. But Pterygotus was not the largest eurypterid, nor even close to the maximum size this group would achieve. That distinction belongs to Jacalopterus rhenanii, discovered in the early Devonian Klurf Formation of Germany. In 2007, paleontologists announced the discovery of a chelicera measuring 46 centimeters long and missing a quarter of its length, meaning the complete claw would have been 45.5 centimeters from base to tip. Using body-to-claw ratios derived from other pterygotid eurypterids where complete specimens are known, researchers calculated that Jacalopterus rhenanii reached a total body length of 2.3 to 2.6 meters, between 7.5 and 8.5 feet. With chelicerae extended forward, the animal would have measured over 11 feet from claw tip to tail. This makes Jacalopterus the largest arthropod ever to have evolved. surpassing even the giant carboniferous millipede Arthropleura. To put this in perspective, Dinkleosteus was longer than most adult humans and far heavier, a predator of dimensions that no arthropod before or since has equaled. The anatomy of these giant eurypterids reveals animals optimized for ambush predation in shallow coastal waters, estuaries and brackish lagoons. Their bodies were segmented and flexible, covered in a scale-like ornamentation that may have provided hydrodynamic efficiency or sensory function. The prosoma, the head shield, was subtrapezoidal with rounded corners, and mounted near the front edges were large compound eyes that provided exceptional visual acuity comparable to modern predatory arthropods. These eyes gave eurypterids the ability to detect movement, judge distances, and track prey with precision. The sixth pair of appendages had evolved into broad swimming paddles, flattened and expanded into structures resembling oars. Unlike their walking legs, which were small and slender and probably not capable of supporting the animal's weight on land, these paddles were robust and muscular, designed for powerful propulsive strokes. Swimming mechanics varied among eurypterid families. Eurypterines like Eurypterus used a drag-based rowing locomotion, moving their paddle appendages synchronously in near horizontal planes. The paddle blades oriented almost vertically on the backward and downward stroke, pushing the animal forward and upward, then rotated horizontally on the recovery stroke to minimize drag. This type of swimming, also used by modern crabs and water beetles, allowed for rapid acceleration. A juvenile Eurypterus just 16.5 centimeters long could achieve 2.5 body lengths per second. Larger adults, meanwhile, likely employed underwater flying or subaqueous flight, in which the sinuous motion and hydrofoil shape of the paddles generated lift, similar to the swimming of sea turtles and sea lions. Though this method had slower acceleration, it was far more energy efficient for large-bodied animals with higher drag coefficients. Adults of Eurypterus could cruise at speeds of 3 to 4 meters per second when using this technique. Pterygatids like Pterygotus and Dinkleosteus, however, possessed a different joint configuration that allowed their paddles to move in more varied planes, suggesting they could swim faster and with greater maneuverability than the more rigid Eurypterines. Their flattened, expanded telsons, the hindmost body segment, served as rudders, providing steering control and stability during pursuit. Trace fossils attributed to eurypterids show trackways and drag marks consistent with benthic movement, indicating that these predators did not swim continuously, but instead cruised near the bottom, using occasional bursts of speed to ambush prey. The combination of large grasping claws, excellent vision, powerful swimming, and patient ambush tactics made pterygatid eurypterids the most formidable predators of their time. Marine equivalents of modern crocodiles, waiting motionless before striking with explosive speed. Eurypterids were not exclusively marine creatures, despite their common name as sea scorpions. Only the earliest members of the group were truly oceanic. Many later forms lived in brackish estuaries, coastal lagoons, and eventually fully freshwater rivers and lakes. This ecological transition from marine to freshwater occurred gradually throughout the late Ordovician to Devonian, driven partly by competition with newly evolving jawed fish and partly by the opportunities presented by expanding freshwater ecosystems that were becoming colonized by plants and invertebrates for the first time. Recent taxonomic analyses suggest that eurypterids accomplished this habitat expansion through a series of incremental steps, with some lineages maintaining populations in both marine and freshwater environments simultaneously, before eventually committing fully to one or the other. The Silurian deposits of New York's Bertie and Salina groups, which preserve thousands of eurypterid fossils including the famous Eurypterus remipes, New York's state fossil, were once interpreted as hypersaline evaporate lagoons where eurypterids tolerated extreme saltwater conditions. But detailed stratigraphic analysis in 2016 revealed that the association of eurypterid fossils with salt hoppers and halite crystals was diagenetic, not ecological. The salt structures formed after burial when evaporating groundwater precipitated minerals around organic remains, not during the animals' lives. The eurypterids themselves likely inhabited normal marine to slightly brackish subtidal waters and were buried during storm events or sea level fluctuations before diagenetic salt crystals grew through their fossils. Direct evidence of eurypterid diet is frustratingly sparse. No gut contents have been definitively identified in any eurypterid fossil, but their anatomy leaves little doubt they were carnivorous. Large size, stereoscopic vision capable of perceiving depth, and appendages covered in spines or equipped with robust claws all point toward predation. Eurypterid coprolites, fossilized feces, from Ordovician deposits in Ohio, contain fragments of trilobites and smaller eurypterids, providing direct evidence that at least some species, such as Megalograptus ohioensis, were active predators and occasional cannibals. Similar coprolites attributed to Lanacopterus dolichosceles contain fragments of jawless fish and smaller specimens of Lanacopterus itself. The specialized appendages seen in different eurypterid families reflect diverse feeding strategies. Carcinosomatoids like Mixopterus and Megalograptus possessed forward-facing appendages with enormously elongated spines, ideal for impaling soft-bodied prey or pinning fast-moving fish against the substrate. Pterygotids like Pterygotus and Jacalopterus had robust chelicerae with serrated inner edges, perfectly designed for crushing and tearing flesh. Biomechanical analyses published in 2022 revealed that different pterygotid species exhibited variable chelicerae morphologies and stress distribution patterns, indicating they targeted different prey types. Some specialized for soft-bodied animals, others for crustaceans with harder cuticles. The most specialized eurypterids were the stylonurine sweep feeders, which independently evolved in two families, the Styloneuroidea and Mycteropoidea, during the Silurian and Devonian. These benthic eurypterids modified their anterior appendages into rake-like structures armed with closely spaced comb-like spines that could be swept through sediment to capture small invertebrates. By comparing the spacing between spines to the feeding structures of modern suspension feeders, researchers calculated that most stylonurines captured prey ranging from 1.6 to 52 millimeters, small benthic crustaceans, mollusks, and worms. But one Mycteropoid family, the Hibbertopteridae, evolved extraordinarily fine spine spacing capable of capturing mesoplankton directly from suspension, making them true filter feeders. This specialization may have been key to their survival during the late Devonian extinction, when most eurypterid groups were decimated. The late Devonian mass extinction, around 375 to 359 million years ago, was catastrophic for eurypterids. Marine eurypterids, which had dominated shallow seas for over 100 million years, were hardest hit. The eurypteran suborder lost over 50% of its diversity in just 10 million years during the early Devonian, and by the late Devonian, only two eurypteran families remained, Adelophthalmidae and Waringopteridae. 9 families went extinct during the Frasnian and Furmenian stages alone. The timing strongly suggests that the rise of jawed placoderm, fish, heavily armored predators like Dunkleosteus, that could crush any prey directly, competed with eurypterids and ultimately displaced them from their ecological roles. Eurypteran decline began precisely when jawless fish became more derived and accelerated with the emergence of placoderms in North America and Europe. By the end of the Devonian, eurypterids had been entirely driven from marine environments and survived only in freshwater habitats, where fish competition was initially less intense. The last eurypterid radiation occurred in the late Devonian and Carboniferous, as Mycteropoid stylonurines expanded into rivers, swamps, and lakes. But this was a diminished relict fauna, a shadow of the group's former glory. Eurypterids persisted in declining numbers through the Carboniferous and into the Permian, finally disappearing entirely in the Permian Triassic. Mass extinction 252 million years ago, the same event that would wipe out 90% of all marine species and reset life on Earth. The Devonian period, spanning from 419 to 359 million years ago, witnessed a transformation so profound that it earned the nickname Age of Fishes. For over 100 million years, vertebrate life had been confined to primitive, jawless forms, lampreys and hagfish relatives that filtered plankton or scavenged dead organisms from the seafloor. But around 430 million years ago, at the end of the Silurian, a revolutionary innovation appeared: hinged jaws. This single anatomical development unlocked an explosion of predatory possibilities, allowing vertebrates to bite, tear, crush, and process prey in ways that soft-bodied filter feeders never could. By the early Devonian, jawed fish had diversified into multiple lineages, including primitive sharks, ray-finned fish, lobe-finned fish, and most significantly, placoderms, the plate-skinned, armored fish that would dominate marine ecosystems for the next 60 million years. Placoderms were unlike any fish alive today. Their heads and anterior bodies were encased in thick, bony armor plates that articulated like medieval plate mail, providing protection while still allowing movement. These plates were not scales but rather dermal bone, mineralized tissue that formed directly in the skin and could reach several centimeters in thickness in the largest species. The armor served multiple functions: defense against other predators, structural support for massive jaw muscles, and possibly ballast for controlling buoyancy. Placoderms ranged in size from just a few centimeters to over 10 meters in length, and they occupied every available niche from shallow reefs to deep ocean basins. Some were bottom-dwelling filter feeders with tiny teeth, others were fast-swimming pursuit predators, and still others were crushing specialists that fed on hard-shelled mollusks and crustaceans. But the undisputed apex predator of the late Devonian seas was Dunkleosteus terrelli, a monstrous placoderm that reached lengths of 20 feet and weighed an estimated 3,600 kilograms, nearly 4 tons. Named after David Dunk, curator of vertebrate paleontology at the Cleveland Museum of Natural History, who spent decades collecting specimens from the Cleveland Shale of Ohio, Dunkleosteus has become an icon of prehistoric predation. Its most distinctive feature was its jaws, or rather, the terrifying substitute for jaws that evolution had crafted. Unlike modern fish with teeth set in sockets, Dunkleosteus possessed elongated, blade-like bony projections that formed from the jawbones themselves. These were not true teeth that could be replaced if broken. They were permanent extensions of the skull, but nature had found an elegant solution to the problem of wear. The edges were self-sharpening. As the upper and lower jaw blades slid past each other during biting, they ground against one another like scissors, maintaining razor-sharp cutting edges throughout the animal's life. Biomechanical studies have revealed that Dunkleosteus possessed one of the most powerful bites in vertebrate history. Using fossil skull reconstructions, finite element analysis, and comparisons with modern predatory fish, researchers calculated that Dunkleosteus could generate bite forces exceeding 6,000 newtons at the front of its jaws, equivalent to roughly 1,350 pounds of pressure. More remarkably, it could open its jaws in as little as 50 milliseconds, creating a powerful suction effect that drew prey into its mouth before the victim could react. This combination of explosive gape speed and crushing bite force made Dunkleosteus one of the most efficient killing machines the oceans had ever seen. It hunted other placoderms, early sharks, bony fish, and possibly even other Dunkleosteus. Fossilized Dunkleosteus bones have been found bearing bite marks that match the spacing and size of Dunkleosteus jaws, suggesting cannibalism was not uncommon. The anatomy of Dunkleosteus reveals an animal supremely adapted for ambush predation in the murky waters of late Devonian seas. Its body was relatively short and stocky compared to modern sharks, with a massive armored head comprising nearly 1/3 of its total length. The armor plating on the head shield alone could weigh several hundred pounds, requiring extraordinary muscle power just to move the head from side to side. Behind the armored section, the body was likely covered in tough, leathery skin rather than scales or additional armor plates. Evidence from fossil articulation patterns suggests the posterior body needed to be flexible for efficient swimming. The tail, though rarely preserved, was probably heterocercal like that of sharks, with the upper lobe longer than the lower, providing lift to counteract the weight of the heavy head. Despite its fearsome reputation, Dunkleosteus faced significant challenges simply existing at its enormous size. The Devonian oceans had lower oxygen concentrations than modern seas, perhaps 15 to 18% of atmospheric levels compared to today's 21%. This would have limited the metabolic rates of large active predators, constraining how fast they could swim and how much energy they could expend hunting. Dunkleosteus likely employed an ambush strategy similar to modern crocodiles, remaining motionless near the seafloor or in kelp forests, conserving energy until prey wandered within striking distance. Then, with explosive acceleration powered by its massive trunk muscles, Dunkleosteus would lunge forward, jaws gaping open to create a vacuum that sucked prey into its guillotine-like bite. The fossil record preserves tantalizing glimpses of Dunkleosteus behavior beyond predation. Fossilized regurgitated masses, essentially fossil vomit, have been found associated with Dunkleosteus remains. These masses contain partially digested bones and armor plates from other placoderms, confirming that Dunkleosteus regularly consumed heavily armored prey. Interestingly, the bones in these masses show minimal digestion, suggesting that Dunkleosteus, like modern sharks, may have expelled indigestible hard parts after extracting nutritious soft tissues. This feeding strategy would have been necessary because Dunkleosteus, despite its formidable jaws, lacked a true stomach and instead had a relatively simple digestive tract more similar to that of lampreys than modern-jawed fish. Dunkleosteus was not alone among giant placoderms. The late Devonian seas teemed with armored predators, each occupying slightly different ecological niches to reduce competition. Titanichthys, a close relative of Dunkleosteus, reached similar lengths but possessed weak jaws with tiny blade-like projections, suggesting it was a filter feeder that used gill rakers to strain plankton from the water column like modern baleen whales. Gorgonichthys, another member of the Dunkleostidae family, had more robust jaw blades than Titanichthys, but remained smaller than Dunkleosteus, possibly specializing in mid-sized prey. Eastmanosteus and Heintsichthys were medium-sized predatory placoderms found in the same formations as Dunkleosteus, their fossils often bearing bite marks from larger predators, likely Dunkleosteus itself. The dominance of placoderms, however, was not destined to last. The late Devonian extinction event, which occurred in pulses between 375 and 359 million years ago, devastated marine ecosystems. The causes were complex and multifaceted. Volcanic eruptions pumped greenhouse gases into the atmosphere, triggering rapid climate oscillations. Shallow seas became choked with algal blooms as nutrient runoff from newly evolved land plants washed into coastal waters, creating anoxic dead zones. Ocean temperatures fluctuated wildly, and sea levels rose and fell repeatedly, fragmenting habitats and isolating populations. Placoderms, as large-bodied predators with specialized diets and slow reproductive rates, were particularly vulnerable. By the end of the Devonian, every single placoderm species had vanished. Dunkleosteus torelli, the apex predator that had ruled the seas for millions of years, disappeared along with its relatives, leaving no descendants. The extinction was so complete that for decades, paleontologists debated whether placoderms were even fish at all. Their bizarre anatomy seemed too alien, too unlike any living vertebrate. Only through cladistic analysis and detailed comparisons of skull structure, jaw articulation, and developmental patterns did scientists confirm that placoderms were indeed jawed vertebrates, occupying a position on the evolutionary tree between jawless fish and modern gnathostomies. The extinction of placoderms opened ecological space for other fish groups that had been suppressed by placoderm dominance. Early sharks, which had existed as relatively minor components of Devonian ecosystems, underwent massive radiation in the Carboniferous period that followed. Bony fish, both ray-finned and lobe-finned, diversified explosively, eventually giving rise to the 30,000-plus species that dominate modern oceans and rivers. And one lineage of lobe-finned fish would take the most audacious step of all, crawling out of the water entirely to colonize land and ultimately giving rise to all terrestrial vertebrates, including the synapsids that would produce the next generation of apex predators. The age of armored fish had ended, but the evolutionary experiments it unleashed would shape the future of life on Earth in ways the placoderms could never have imagined. The Carboniferous period, lasting from 359 to 299 million years ago, was a time of extraordinary transformation on Earth. For the first time in planetary history, vast forests stretched across the continents, their towering lycopsid trees, scale trees, reaching heights of 100 feet or more. These were not the forests we know today. They were strange, humid jungles dominated by tree-sized club mosses, horsetails, and early ferns that created dense, swampy canopies. Beneath them, the ground was a waterlogged morass of decaying plant matter that would eventually become the coal deposits from which the period takes its name. But the most remarkable feature of the Carboniferous was invisible. The atmosphere itself had become a biological accelerant. Oxygen levels, driven upward by the photosynthetic output of millions of square miles of vegetation, and limited by the slow evolution of wood-decomposing fungi, reached concentrations never seen before or since. At its peak, atmospheric oxygen climbed to approximately 35%, nearly two-thirds higher than the 21% we breathe today. This oxygen-rich air had profound consequences for animal life, particularly for arthropods whose respiratory systems were uniquely positioned to exploit the surplus. Insects had colonized land during the Devonian, initially as small, wingless forms that scavenged organic debris and hunted tiny prey. But by the early Carboniferous, some lineages had evolved flight, the first animals ever to take to the air. This innovation opened an entirely new dimension of ecological space, free from terrestrial predators and rich with flying insects to prey upon. And in the oxygen-saturated atmosphere, these early flyers grew to sizes that defy modern comprehension. The most iconic was Meganeura monyi, often called a giant dragonfly, though it was actually a member of an extinct order called Meganosoptera, the griffin flies. Meganeura possessed a wingspan that reached 65 to 75 centimeters, over 2.5 feet from wingtip to wingtip, making it larger than many modern seabirds. With its enormous multifaceted eyes, powerful serrated mandibles, and spiny legs designed for grasping prey in mid-air, Meganeura was an aerial predator of devastating efficiency. The reason insects could achieve such gigantic proportions in the Carboniferous lies in the fundamental constraints of their respiratory system. Unlike vertebrates, which use lungs and circulatory systems to distribute oxygen via hemoglobin in blood, insects rely on a network of tubes called tracheae that deliver oxygen directly to tissues through passive diffusion. Air enters through openings called spiracles and travels through increasingly narrow tubules until it reaches individual cells. This system works brilliantly at small scales. It's one reason insects are so successful today, but it imposes strict size limits. As body size increases, the distance oxygen must diffuse also increases. And because diffusion rates depend on concentration gradients, there comes a point where cells deep inside the body simply cannot receive enough oxygen to sustain metabolism. Modern insects are constrained by this limitation to relatively modest sizes, with the largest flying insects today, such as certain hawk moths and atlas moths, having wingspans of just 10 to 12 inches. But in the Carboniferous, the rules changed. With oxygen concentrations 66% higher than modern levels, the partial pressure of oxygen in the atmosphere was proportionally greater, steepening the concentration gradient and allowing diffusion to occur more rapidly and over longer distances. This meant that insects could grow substantially larger before hitting the diffusion limit. Mathematical models and physiological experiments with modern insects in hyperoxic environments have confirmed this principle. When oxygen availability increases, maximum sustainable insect size increases proportionally. The effect was most pronounced in active flying insects like Meganeura, which required high metabolic rates to power flight muscles. The combination of passive diffusion enhancement and reduced metabolic constraints allowed griffinflies to dominate Carboniferous skies in a way no insect has since. Meganeura was not merely large, it was a superbly adapted aerial hunter. Its 4 wings were independently articulated, allowing for exceptional maneuverability. Griffinflies could hover, dart sideways, and even fly backward, much like modern dragonflies, but with far greater power and speed. Each wing was reinforced by a complex network of veins that provided structural rigidity while remaining light enough for efficient flight. The compound eyes, among the largest of any insect, provided nearly 360-degree vision, enabling Meganeura to detect prey from almost any angle. Fossil evidence and comparisons with living relatives suggest that Meganeura was an ambush predator, perching on tree trunks or hovering in forest clearings, then launching itself at passing insects with explosive acceleration. Its prey included smaller flying insects, early terrestrial arthropods, and possibly even juvenile amphibians that ventured too close to the water's edge. The wings themselves reveal sophisticated aerodynamic design. Each possessed a prominent nodus, a notch along the leading edge, that acted as a hinge, allowing the outer portion of the wing to twist independently during the stroke cycle. This feature, still present in modern dragonflies, generates additional lift and improves efficiency at high speeds. The wing membrane was reinforced by sclerotized patches that prevented tearing during violent maneuvers, and microscopic analysis of fossilized wings has revealed fine corrugations that likely increase structural strength without adding weight. Meganeura's thorax was dominated by massive flight muscles, occupying perhaps 40% of the animal's total body mass, capable of generating the wing beat frequencies necessary for sustained flight, despite the creature's substantial size. Estimates suggest that Meganeura could achieve flight speeds of 15 to 20 miles per hour, faster than most modern dragonflies, and more than sufficient to overtake any prey insect of its era. But Meganeura did not hunt alone in the Carboniferous skies. The atmosphere teemed with a diverse assemblage of giant flying insects, many of which have no modern analogues. Protodonate dragonflies of the family Gyroptera rivaled Meganeura in size, with some species possessing wingspans approaching 60 centimeters. Early mayflies, though less predatory, also reached impressive dimensions, their delicate wings spanning 15 to 20 centimeters compared to the few centimeters typical of modern species. Even more bizarre were the Paleozoic, an extinct order of beaked insects that combined features of modern cicadas and moths. Some Paleozoic had wingspans exceeding 40 centimeters and possessed elongated rostrum-like mouthparts that they likely used to pierce plant tissues and feed on sap. These herbivorous giants played critical roles in Carboniferous ecosystems, serving both as primary consumers that controlled plant growth and as prey for carnivores like Meganeura. The forests in which these giant insects lived were unlike any modern ecosystem. The climate was warm and humid year-round, with little seasonal variation in the equatorial regions where most coal swamps developed. Rainfall was abundant, and the flat topography combined with poor drainage created vast wetland systems that stretched for hundreds of miles. The dominant trees, Lepidodendron, Sigillaria, and Calamites, had shallow root systems adapted for waterlogged soils, and their bark was covered in diamond-shaped leaf scars that gave them a distinctive reptilian appearance. These trees grew rapidly, but had low-density wood with minimal structural lignin, making them prone to toppling during storms. When they fell, they accumulated in oxygen-poor water where decomposition was slow, and over millions of years, this organic matter was compressed and transformed into the coal seams that powered the Industrial Revolution. The undergrowth was a tangle of ferns, seed ferns, and primitive horsetails, creating a dense, multi-layered habitat. Sunlight penetrating to the forest floor was limited, and the air beneath the canopy was thick with moisture and the smell of rotting vegetation. Pools of stagnant water dotted the landscape, breeding grounds for aquatic insect larvae and the amphibians that fed on them. It was in this humid, oxygen-saturated world that arthropods reached their maximum terrestrial sizes, not only in the air, but also on the ground, where even more astonishing giants lurked. While Meganeura ruled the skies, the forest floor of the Carboniferous harbored arthropods of even more staggering dimensions. The undergrowth teemed with oversized centipedes, enormous scorpions, and spiders larger than dinner plates. But the undisputed champion of terrestrial arthropod gigantism was Arthropleura, a millipede relative that stretched up to 2.5 meters, over 8 feet in length, and weighed an estimated 50 kilograms, roughly 110 pounds. To put this in perspective, Arthropleura was longer than most adult humans, and as heavy as a large dog, making it the largest terrestrial invertebrate ever to have evolved. Fossils of Arthropleura have been found in coal deposits across North America and Europe, particularly in Scotland, England, Germany, and the eastern United States. The creature's fossils are distinctive. Segmented body plates with a characteristic ribbed texture, massive leg attachments, and trackways preserved in fossilized mud that show the animal's gait and movement patterns. Unlike modern millipedes, which are primarily detritivores feeding on decaying plant matter, Arthropleura's ecology remains debated. Some paleontologists argue it was an herbivore, using powerful mandibles to process tough plant tissues, including early seed ferns and primitive conifers. Others suggest it may have been omnivorous or even predatory, capable of overpowering smaller arthropods and amphibians with its sheer size and strength. Fossilized gut contents have not been definitively identified, leaving the question open. What is certain is that Arthropleura possessed formidable anatomy. Its body consisted of 30 or more armored segments, each bearing a pair of powerful legs that moved in coordinated waves. The exoskeleton was reinforced with calcium carbonate, providing both structural support and protection from predators. The head bore compound eyes and sensory antennae, and the mouthparts, though not preserved in most specimens, were likely robust and capable of processing tough food items. Arthropleura's immense size, like that of Meganeura, was made possible by the hyperoxic carboniferous atmosphere. Millipedes and their relatives breathe through spiracles and tracheal systems similar to insects, and they face the same diffusion-based size constraints. In modern environments, even the largest millipedes rarely exceed 30 centimeters in length, constrained by oxygen availability. But in the Carboniferous, with oxygen at 35%, diffusion rates were sufficient to support bodies 10 times longer and hundreds of times more massive. The abundance of plant matter in coal swamp forests provided ample food resources, and the lack of large terrestrial vertebrate predators. Most amphibians were confined to aquatic or semi-aquatic lifestyles, meant that size was not a liability, but rather an advantage. Larger arthropods could access food sources unavailable to smaller competitors, intimidate potential threats, and maintain higher body temperatures through thermal inertia, improving metabolic efficiency in cool nighttime conditions. The coal swamps were also home to giant scorpions, some reaching lengths of 70 centimeters. These were among the first arachnids to fully adapt to terrestrial life, having evolved book lungs that allowed them to breathe air efficiently. Carboniferous scorpions retained many features of their aquatic ancestors: thick, heavily armored bodies and large pincers. But they had fully functional walking legs and venom-injecting stingers, making them formidable predators. They likely hunted smaller arthropods, early terrestrial vertebrates, and perhaps even juvenile Arthropleura, ambushing prey from beneath logs or in burrows. Spiders, too, reached impressive sizes during this period. Though none approached the dimensions of Arthropleura, some Carboniferous spiders had leg spans exceeding 20 centimeters and possessed silk glands capable of producing webs strong enough to trap large flying insects like griffin flies. The evolutionary arms race between aerial predators and ground-dwelling hunters drove increasing specialization. Some spiders developed trapdoor burrows to ambush passing arthropods, while others built extensive sheetwebs in the understory. But the age of giant arthropods was not destined to last. As the Carboniferous gave way to the Permian period around 299 million years ago, atmospheric oxygen levels began to decline. The evolution of lignin-degrading fungi allowed dead plant matter to decompose more rapidly, reducing the burial of organic carbon and consequently decreasing oxygen production. Simultaneously, the climate began to shift toward cooler, drier conditions as the continents coalesced into the supercontinent Pangaea. The vast coal swamps dried up and were replaced by more arid environments dominated by conifers and early reptiles. As oxygen levels dropped toward modern concentrations, the physiological advantages that had supported gigantic arthropods disappeared. Arthropleura and its kin gradually declined, and by the end of the Permian, giant terrestrial arthropods had vanished entirely, replaced by an entirely new caste of apex predators, the synapsids, mammal ancestors that would redefine what it meant to be a terrestrial carnivore. The Permian period, stretching from 299 to 252 million years ago, marked a fundamental transition in the history of terrestrial life. The humid coal swamps of the Carboniferous had given way to seasonal, often arid climates as the supercontinent Pangaea consolidated. With this environmental shift came a dramatic turnover in fauna. Amphibians, which had dominated Carboniferous ecosystems, were increasingly restricted to waterways and wetlands, while a new group of vertebrates, the synapsids, rose to prominence on land. Synapsids were not reptiles, though they superficially resembled them. They belonged to an entirely separate evolutionary lineage, one that would eventually give rise to all mammals, including humans. The earliest synapsids, collectively known as pelycosaurs, appeared in the late Carboniferous, but truly flourished in the early Permian, diversifying into a remarkable array of herbivores, omnivores, and carnivores. Among the most iconic pelycosaurs was Dimetrodon, a predator whose name means 2 measures of teeth in reference to its differentiated dentition. Dimetrodon reached lengths of up to 4.6 meters, over 15 feet, making it one of the largest land predators of its time. But its most distinctive feature was the enormous sail-like structure rising from its back, supported by elongated neural spines that projected upward from the vertebrae. Some individuals bore sails that stood nearly a meter tall, creating a dramatic profile that has made Dimetrodon one of the most recognizable prehistoric animals. For over a century, scientists debated the function of this sail. Early hypotheses suggested it served as a thermoregulatory device, a biological solar panel that absorbed heat in the morning to warm the animal's blood, allowing it to become active earlier than competitors. Computer models and vascular analyses of the sail structure revealed networks of blood vessels that could have facilitated heat exchange, supporting this interpretation. However, alternative explanations have gained traction in recent decades. The sail may have functioned primarily as a display structure used in intraspecific competition and mate selection, similar to the crests of modern birds or the horns of ungulates. Sexual dimorphism in sail size among fossil specimens suggests that males may have possessed larger, more elaborate sails to intimidate rivals and attract females. The sail could also have served multiple functions simultaneously: thermoregulation during cool mornings, display during mating season, and perhaps even as a fat storage hump during times of plenty, with the sail providing structural support. Whatever its precise function, the sail was costly to maintain, requiring significant calcium and phosphorus to build the elongated vertebral spines, and it made Dimetrodon more visible to both prey and potential threats. The fact that natural selection favored this structure for millions of years suggests it provided substantial adaptive benefits. Dimetrodon was a formidable predator, equipped with a skull that measured up to 40 centimeters in length and jaws lined with sharp recurved teeth. Unlike reptiles, which possess relatively uniform conical teeth, Dimetrodon had differentiated dentition. Large canine-like teeth for puncturing and gripping, smaller incisors for snipping, and serrated posterior teeth for shearing flesh. This dental specialization is a synapomorphy, a shared derived characteristic of synapsids, and one of the features that would eventually lead to the complex tooth arrangements seen in mammals. Dimetrodon's jaw muscles were also more sophisticated than those of contemporary reptiles, with attachment points on the skull that allowed for a powerful, sustained bite. Bite force estimates suggest Dimetrodon could generate pressures sufficient to crush bone, making it capable of taking down large prey, including early herbivorous synapsids and reptiles. But Dimetrodon was not alone in the early Permian. The terrestrial ecosystems of this period supported a complex web of pelycosaur predators and prey. Edaphosaurus, a close relative of Dimetrodon, also possessed a sail, but was entirely herbivorous, using blunt, peg-like teeth to process plant material. Ophiocodon was a semi-aquatic carnivore that hunted fish and amphibians in rivers and lakes. Sphenocodon, a smaller predator without a sail, likely pursued fast-moving prey through the underbrush. These pelycosaurs represented the first radiation of large terrestrial vertebrates, pioneering ecological roles that would be refined by their descendants. By the Middle Permian, around 270 million years ago, pelycosaurs were in decline, gradually replaced by more derived synapsids called therapsids. Therapsids possessed more upright limb postures, more sophisticated jaw musculature, and evidence of higher metabolic rates. They diversified rapidly into two major groups: the herbivorous decinodonts, which would become the most abundant land vertebrates of the late Permian, and the carnivorous theriodonts, which included the ancestors of mammals. Among the theriodonts, one lineage would evolve the most fearsome predators the Permian world had ever seen: the gorgonopsids, saber-toothed killers that would reign supreme until the greatest catastrophe in Earth's history erased them forever. In the late Permian, between 260 and 252 million years ago, a group of therapsid predators evolved that prefigured the saber-toothed cats of the Cenozoic era by over 200 million years. These were the gorgonopsids, named after the Gorgons of Greek mythology for their fearsome appearance, and they represented the pinnacle of pre-mammalian predation. Gorgonopsids ranged dramatically in size, from species no larger than modern house cats to giants with skulls exceeding 60 centimeters in length and total body lengths approaching 3.5 meters. The largest and most formidable was Inostrancevia alexandri, discovered in Russia's late Permian deposits and named after geologist Alexander Inostrantsev. With a skull measuring up to 60 centimeters and massive saber-like canine teeth reaching 15 centimeters in length, Inostrancevia was the apex predator of its ecosystem, capable of taking down prey far larger than itself. The defining feature of gorgonopsids was their dentition. Unlike the relatively uniform teeth of reptiles, or even the differentiated teeth of earlier pelycosaurs like Dimetrodon, gorgonopsids possessed dramatically enlarged canine teeth that protruded from the upper jaw like daggers. These sabers were laterally compressed and often featured serrated edges on the anterior and posterior margins, designed explicitly for slicing through flesh and severing blood vessels. The canines grew continuously throughout life to compensate for wear, a feature also seen in modern saber-toothed predators. But perhaps most remarkably, gorgonopsids could open their jaws to angles approaching 90 degrees, far wider than any modern mammal, allowing them to bring their massive canines to bear even when grappling with large, struggling prey. Recent histological studies have revolutionized our understanding of gorgonopsid biology. In 2024, researchers examined the bone microstructure of multiple gorgonopsid specimens, including Inostrancevia, using thin sections and polarized light microscopy. The analysis revealed densely vascularized fibrolamellar bone, a tissue type associated with rapid growth and high metabolic rates. This finding suggests that gorgonopsids grew quickly during their early years, reaching adult size in just a few years rather than the decades required by similarly sized reptiles. The bone structure also contained growth marks analogous to tree rings, allowing researchers to estimate individual ages at death. Surprisingly, Some gorgonopsid individuals lived for 10 to 15 years or more, considerably longer than previously assumed for such active predators. The combination of fast growth and relatively long lifespans indicates that gorgonopsids had elevated metabolic rates intermediate between modern reptiles and mammals. They were likely mesothermic, maintaining body temperatures higher than the environment, but not fully endothermic like modern mammals. Gorgonopsid anatomy reveals animals built for explosive power rather than endurance pursuit. Their limbs, though more upright than those of Paleozoics, still sprawled somewhat laterally, suggesting they moved with a semi-erect gait similar to modern crocodiles. The forelimbs were robust and heavily muscled, with powerful claws capable of grasping and pinning prey. The hind limbs were longer and more gracile, providing thrust during ambush attacks. Skeletal reconstructions suggest gorgonopsids were capable of short bursts of speed, perhaps 20 to 25 miles per hour, but could not sustain high velocities for long distances. This is consistent with an ambush hunting strategy. Gorgonopsids likely stalked prey through the coniferous forests and fern prairies of the late Permian, relying on stealth and sudden acceleration to close the distance before delivering a devastating bite to the neck or flank. The skull of gorgonopsids was massively constructed, with broad zygomatic arches that housed enormous temporalis muscles, the primary jaw-closing muscles. The jaw joint was positioned below the level of the tooth row, creating a mechanical advantage that allowed gorgonopsids to generate tremendous bite forces despite their relatively gracile mandibles. The canines themselves were not used for crushing or bone breaking, but rather for piercing and slashing. Gorgonopsids would sink their sabers deep into prey, then tear laterally to open massive wounds. This technique, similar to that employed by modern big cats, caused rapid blood loss and shock, quickly incapacitating even large, heavily built herbivores. The late Permian world in which gorgonopsids lived was a harsh seasonal environment dominated by the supercontinent Pangaea. The interior of the landmass was arid, with vast deserts and scrublands broken by seasonal rivers. But along the southern margins, in what is now South Africa, Russia, and parts of Antarctica and South America, more temperate zones supported diverse ecosystems. These regions were home to massive dicynodont herbivores, barrel-bodied beaked synapsids that grazed on tough vegetation. Some dicynodonts, such as moschops, weighed over a ton and traveled in herds for protection. These were the primary prey of gorgonopsids like Inostrancevia, which likely followed herds and picked off isolated, sick, or young individuals. But despite their success, gorgonopsids and the entire late Permian ecosystem were living on borrowed time. Approximately 252 million years ago, the Earth was struck by the most catastrophic extinction event in its history, the Permian Triassic extinction, often called the Great Dying. This event would erase 57% of all families, 81% of marine species, and an estimated 70% of terrestrial vertebrate species, including every single gorgonopsid. The saber-toothed killers that had dominated the land for 10 million years would disappear completely, their ecological roles empty for millions of years until new predators evolved to fill them. 9 million years ago, the Earth underwent a transformation so rapid and so complete that it nearly ended complex life entirely. The Permian Triassic extinction event, the Great Dying, was the most severe biodiversity crisis in recorded geological history, far surpassing the asteroid impact that killed the dinosaurs. In the oceans, an estimated 81 to 96% of all marine species vanished. On land, 70% of terrestrial vertebrate species disappeared, including all gorgonopsids, most dicynodonts, and the last pelycosaurs. Insects, which had survived every previous extinction event virtually unscathed, suffered the only mass extinction in their 400 million year history, with 8 orders going completely extinct. Forests collapsed worldwide, replaced by fern-dominated landscapes devoid of tree cover. The oceans became toxic, oxygen-depleted dead zones where only a handful of generalist species could survive. It would take 10 million years, longer than any other extinction, recovery, for ecosystems to regain even a fraction of their former complexity. The proximate cause of the Great Dying was catastrophic volcanism on an almost unimaginable scale. In what is now Siberia, a mantle plume breached the Earth's crust and triggered one of the largest volcanic events in planetary history, the Siberian Traps eruptions. Over the course of perhaps just 60,000 years, magma covered an area of 7 million square kilometers, larger than the contiguous United States, with lava flows up to 6 kilometers thick in places. The volume of magma erupted exceeded 4 million cubic kilometers, making it one of the most massive volcanic events ever documented. But the direct destruction from lava flows was trivial compared to the atmospheric and climatic havoc the eruptions unleashed. The magma intruded through coal seams and evaporate deposits, vaporizing vast quantities of hydrocarbons and releasing unprecedented amounts of carbon dioxide, methane, sulfur dioxide, and chlorine into the atmosphere. Within thousands of years, atmospheric carbon dioxide concentrations may have increased tenfold, triggering runaway greenhouse warming that raised global temperatures by 8 to 10 degrees Celsius or more. The oceans, absorbing much of this excess CO2, became acidic. pH levels may have dropped by 0.7 units, approaching the acidity of vinegar. Marine organisms with calcium carbonate shells and skeletons simply dissolved. Coral reefs, which had taken millions of years to build, vanished entirely and would not reappear for 10 million years. The warming oceans stratified, with hot surface waters preventing the mixing of oxygen-rich surface layers with nutrient-rich deeper layers. Anoxia spread from the deep ocean upward, eventually reaching the photic zone and killing photosynthetic organisms. Worse still, sulfur-reducing bacteria thrived in these anoxic conditions, producing hydrogen sulfide, a deadly poison that dissolved into seawater and eventually bubbled into the atmosphere, poisoning terrestrial life. On land, the effects were equally catastrophic. The combination of extreme heat, acid rain, toxic gases, and collapsing food webs created an ecological death spiral. Gorgonopsids and other large predators, sitting at the top of food chains, were particularly vulnerable. They required abundant prey populations to survive, and as herbivore diversity collapsed, so did the carnivores that depended on them. Bone histology studies of theropsids that survived the extinction reveal dramatic changes in growth rates and life history strategies. Pre-extinction theropsids showed relatively slow growth and long lifespans, indicating stable, predictable environments. Post-extinction survivors, by contrast, exhibited rapid growth, early sexual maturity, and short lifespans, adaptations for living in chaotic, unpredictable conditions where population crashes were frequent. These survivors were also significantly smaller, with body masses averaging just 10 to 15% of pre-extinction forms. The principle known as the Lilliput effect, the temporary dwarfing of organisms following mass extinctions, is clearly evident. In early Triassic fossils, the extinction unfolded in two main pulses, separated by roughly 60,000 years. The first pulse, coinciding with the initial eruptions of the Siberian Traps, eliminated primarily tropical marine organisms and large terrestrial predators. The second pulse, possibly triggered by a secondary phase of volcanism or by runaway greenhouse feedback mechanisms, was even more severe, wiping out most of the species that had survived the initial crisis. By the end, only a handful of disaster taxa, opportunistic species capable of exploiting degraded environments, remained. On land, the early Triassic was dominated by a single herbivore genus, Lystrosaurus, a pig-sized decinodont that survived by virtue of its burrowing lifestyle and low metabolic requirements. Lystrosaurus fossils comprise an astonishing 95% of all terrestrial vertebrate remains from the earliest Triassic, a level of ecological dominance never seen before or since. The Gorgonopsids, for all their power and sophistication, left no descendants. They were evolutionary dead ends, experiments in predation that flourished for 10 million years before vanishing completely. Their ecological role as apex predators would remain vacant for nearly 10 million years, until Archosaurian reptiles, the ancestors of crocodiles and dinosaurs, evolved to fill the gap. The theropsid lineage itself survived barely, through small generalized forms that would eventually give rise to true mammals in the Jurassic. But the world they inherited was unrecognizable. A hot, desolate wasteland where survival, not dominance, was the only measure of success. In the aftermath of the Great Dying, the Earth slowly began to heal. The Siberian Traps eruptions ceased, and volcanic gases gradually dissipated from the atmosphere. Carbon dioxide levels, though still elevated, began to decline as weathering and biological processes locked carbon back into rocks and sediments. Ocean circulation resumed, and oxygen concentrations slowly recovered. But the recovery was agonizingly slow. It would take 5 to 10 million years for biodiversity to approach even half its pre-extinction levels. The early Triassic world was dominated by disaster taxa, opportunistic, fast-reproducing species that could exploit the ecological wreckage, but lacked the specialization to build complex ecosystems. The theropsid survivors were small, unspecialized forms, many no larger than modern rats. They possessed generalized dentition, omnivorous diets, and rapid reproductive rates that allowed them to persist in unstable environments. These humble creatures bore little resemblance to the mighty gorgonopsids that had preceded them, but they carried within their genomes the innovations that would eventually define mammals. Differentiated teeth, more efficient jaw articulation, possible lactation precursors, and rudimentary endothermy. Over the next 50 million years, these lineages would slowly diversify, giving rise to the cynodonts, dog-toothed theropsids that included the direct ancestors of mammals. By the late Triassic, some cynodonts had evolved fur, advanced thermoregulation, and complex social behaviors. And in the Jurassic, the first true mammals appeared. Tiny nocturnal creatures that lived in the shadows of the dinosaurs. The innovations pioneered by Paleozoic predators did not disappear. They were refined and passed down through evolutionary lineages. The differentiated dentition of Dimetrodon became the heterodonty of mammals, allowing for specialized processing of food. The upright limb posture experimented with by gorgonopsids reached its apex in mammals and dinosaurs, enabling efficient terrestrial locomotion. The compound eyes of trilobites and eurypterids established the template for arthropod vision that persists today. Even the gigantism of carboniferous insects left a legacy. It demonstrated that atmospheric chemistry can fundamentally reshape the rules of animal design, a lesson relevant as we confront modern changes to Earth's atmosphere. Yet these ancient predators remain obscure in popular imagination, overshadowed by dinosaurs and Ice Age mammals. Few people have heard of Anomalocaris, Dunkleosteus, or Inostrancevia, despite their comparable or superior size and ferocity. This obscurity reflects a bias in both scientific research and public outreach. Mesozoic and Cenozoic fossils are more abundant, better preserved, and easier to study than Paleozoic remains. Soft-bodied organisms like Anomalocaris require exceptional preservation and are found only in rare Lagerstätten. Eurypterid fossils, though more common, are often fragmentary, making reconstruction challenging. Gorgonopsid skeletons are known primarily from southern Africa and Russia, regions with limited paleontological infrastructure relative to North America and Europe. But the significance of these creatures extends beyond their individual stories. They demonstrate that the history of predation is older, more diverse, and more complex than commonly appreciated. They reveal that ecosystems have repeatedly assembled, thrived, and collapsed over geological time, each iteration experimenting with new solutions to the challenges of survival. And they remind us that even the most dominant predators are vulnerable to environmental change. Gorgonopsids ruled the late Permian for 10 million years, yet they disappeared entirely when volcanism altered atmospheric chemistry. Placoderms dominated Devonian seas for 60 million years, Yet they vanished when fish with more efficient jaw mechanics outcompeted them. Eurypterids terrorized Silurian waterways for 150 million years, yet they were driven to extinction by the very vertebrates they once hunted. The lesson is clear. No matter how well adapted, how powerful, or how successful a predator becomes, it exists at the mercy of environmental stability. When atmospheres change, oceans acidify, or climates shift, even apex predators can vanish, leaving behind only fossils and the faint imprint of their innovations in the genomes of their descendants. Today, as we face our own period of rapid environmental change, the ancient predators of the Paleozoic offer a sobering reminder. Extinction is not an abstract concept confined to deep time. It is a recurring process that has reshaped life repeatedly, and the creatures we know today, ourselves included, are merely the latest inheritors of a world built on the ruins of countless vanished ecosystems. The ancient predators may be gone, but their story is not finished. It continues in every mammal that walks the earth, every insect that takes to the air, and every ecosystem that assembles, thrives, and one day, inevitably, transforms into something new.